Hi, good morning to everyone and all the presenters today. So, my topic for this morning is based on stimulation of ovarian development and spawning in giant freshwater prawn, Macrobrachium rosenbergi. So, this topic was obtained and cited from a journal paper by Tan Buntek et al. In 2008. So, giant freshwater prawn is a well-known as udang galah in our country, and most Asians love to eat udang galah. And I am pretty sure everyone is familiar with how udang galah looks like, just like in the photo shown here. And the left photo shows you the larva stage of udang galah, and basically, these baby prawns can be found living in the brackish water, and once it matures into adult, it will go back to the freshwater. So in Malaysia, the giant freshwater prawn, Macrobrachium rosenbergi, is becoming an increasingly important targeted species with potential to raise income among impoverished farmers. Because today's topic is about ovarian development, we first must know what is the function of ovaries, and pretty sure everyone already know the answer, which is for reproduction. Even in human females, we have our own ovaries which functions as cells to develop into embryo. It is the same situation as prawns. They have different stages in ovarian development, starting from the immature stage until it becomes fully developed and matured. And it has several stages, such as in this slide, I'm showing you 6 stages of ovarian development, which is adapted from a paper by Huang et al. In 2010. So the first stage is the most immature development of ovary, and the appearance is more like thin, small, rounded, and semi-transparent. Also, we can see that the size is the smallest than the other stages. Stage 2, the size and thickness of the ovary begin to increase a bit, and the color is not semi-transparent anymore. It has yellowish to creamy white coloration. And then in stage 3, it contains green spots on the ovary and they have lobes. Then in stage 4, the color is more like emerald and the size is much bigger than the previous stage. After all, when it reaches stage 5, it becomes matured and we can see the presence of visible oocytes inside it. The size is always bigger as it covers the entire dorsal portion of the hepatopancreas. And the last stage is basically after ovulation and causes the size of ovary to be reduced and thinner. So now we have fully understood how ovaries in prawns are developed. So what are the importance of this process? Of course, first, it determines female prawn's fertility, and then it induces development of eggs and embryos, and the last one also, of course, it increased number of the offsprings. Indirectly, as we obtain more offsprings by inducing these processes, we can increase the market size of udang galah as well as to improve aquaculture industry. So in this paper, it was highlighted that there are 2 chemicals responsible for the development of ovaries in prawn. The first one is known as serotonin, which stimulates production of gonad-stimulating hormone, and octapamine or dopamine, which induces production of gonad-inhibiting hormone. Basically, serotonin induces the release of GSH from the brain and thoracic ganglia. Meanwhile, octapamine acts as antagonist of serotonin by blocking the release of GSH. Note that GSH is important to initiate reproduction processes in the prawns. Here are the aquaculture techniques used to study ovaries development. The first is maintenance and feeding. The mature giant freshwater prawns were maintained at 26 to 27 degrees Celsius in aerated water, and the prawns are fed with pellets and mussels. And then it proceeds to mating and spawning. The females were subjected to mating with the males and spawning happens. After spawning happens, they remove the eggs from the abdomen of the females, and then the females have immature ovaries which are used further for the study. So the females with immature ovaries were injected with 1 to 3 doses of serotonin and octapamine in the abdomen, and then after 10 days, the experiments were basically terminated, and then they observed the ovarian stage based on size and color, and the weight of the prawns were also recorded. Then this study also highlighted how they study about spawning induction, first by injection of serotonin or octapamine, and then it allows the females to mate with the males, and then they record the spawning time, which is followed by the hatchment of the eggs. Based on the study of ovaries development, the author highlighted that ovarian development is stimulated by injection of serotonin, and one dose of serotonin can produce 10% matured ovaries, which develop into stage 3 even after 10 days only. And basically, the octapamine inhibits development of ovaries, while 3 doses were not necessary to induce ovarian development. And the last thing that they highlighted is that controls develop immature stage 1 or developing stage 2 only. The study on spawning induction highlighted that both serotonin and octapamine were able to induce spawning. Treated females were able to spawn faster than the controls, and treated females also spawned most during daytime while controls were at night. Serotonin or octapamine can induce spawning within few hours after mating. One dose of serotonin can induce ovarian development, and later injected with another serotonin or octapamine that can stimulate spawning after pre-mating molt. That's all for my presentation today. Thank you.